I'm Daniel Levine, and this is the Bio Report. Animal testing for experimental therapies is slow, expensive, and an imperfect predictor for how a drug will act in humans. The Foundation for the National Institutes of Health in July announced a program to advance innovative laboratory technologies that model human biology, enabling faster, less expensive testing. We spoke to Stacey Adam, Vice President of Scientific Partnerships for the FNIH and leader of the Public-Private Partnership, about the new program, the technologies being explored, and what it will take to transform biomedical research with better disease models. Stacey, thanks for joining us.
Thanks, Danny. I appreciate you having me.
We're going to talk about the changing landscape for the use of animal testing for experimental therapies, the Foundation for the National Institutes of Health, and its efforts to advance new technologies that can provide an alternative to the use of animal testing of drugs. Animal testing was long required by the U.S. Food and Drug Administration to help establish the safety of new therapies. There have also long been ethical concerns about this, and the FDA earlier this year began to phase out mandatory animal testing as part of a preclinical drug safety studies. This followed 2022 legislation that allowed the agency to accept data from new methodologies. Ethics aside, there are many other problems with animal testing. How reliably does animal testing predict the safety of drugs?
I mean, that's a great question. Since, animal testing has been required probably since the late 30s, numerous drugs have actually still been pulled from the market. So, you know, due to toxicity, even after they've been approved for initial efficacy. So that would lead you to believe that the animal testing that had been done earlier on, not always as predictive as we would have wanted it to be. I mean, even still to this day, with all the preclinical testing that is done, 90% of all drugs still fail in trial. So clearly we need better prediction than what we already have.
At the same time, there are lots of concerns about improving the cost and time of developing new therapies. What role might alternatives to animal testing play in improving R&D efficiency?
Some studies predict that NAMs could actually save the industry over 3 billion a year. I've seen lots of other numbers on that. I think that's a more recent one from a Nature study that had been done around specifically organs on a chip. So that's not an insignificant amount of funds when you look at that for the industry overall. And when you talk about moving timelines from days to weeks instead of weeks to years, the way it is for some animal testing that we can now do with the new approach methodologies, that's only bound to, assist in kind of bringing down costs because you're not paying for labor over that time. You're not paying for the expensive ability to house and continue to kind of keep animals, at the stature that they need to be. In addition, the high throughput screening that is encompassed in a lot of new approach methodologies will allow more compounds to be tested against actual human-based models. So organoids on a chip, whatever kind of, is your flavor of NAM that you prefer. They can do this faster and at a much more reduced cost than trying to scale up the same type of, bulk testing within animal models, even, kind of the more standard animal models of rodents or anything else. It's still going to be far less costly. And I would imagine, hopefully much higher fidelity to human biology, allowing us to have less of those drug candidates that I mentioned, under the first question to fail.
And you use the term NAMs just for listeners, this is new approach methods. This is any alternative to animal testing.
Yes, sorry. New approach methodologies encompass quite a wide variety of types. So, some people go as far back as, cell lines in two-dimensional culture. Some would argue that it's when you start to get to 3D organoids and organs on a chip. But really, NAMS has a very broad category. Those are just the ones that people think about a lot, which are what we consider in vitro NAMS. We also have in silico NAMS. This comes to AI technology, kind of in silico testing of other types, where you can run model systems and that sort of thing. And then even in chemico, which is, think about that as like chemical modeling and different types of things that are done. I think that's probably one of the least known for some folks, unless you kind of work in that area of the field.
The Foundation for the National Institutes of Health is a independent nonprofit established by Congress. For listeners not familiar with it, can you explain how it's funded and the role it plays in supporting the NIH and driving public private partnerships?
Absolutely. So the Foundation for the National Institutes of Health is, you know, for all intents and purposes, a standalone nonprofit. We're a 501c3 registered and recognized in the state of Maryland. But we were brought into an existence by congressional legislation in the mid 1990s. And that makes us a bit unique that we get to work alongside NIH in a very special manner. Most folks don't realize that many of the uh, scientific organizations within the government have a congressionally legislated nonprofit, CDC Foundation, Reagan Udall for FDA, that also help their agencies the same way that we help NIH. And in truth, our entire mission of our organization is to serve the mission of NIH. We amplify what they do through, helping them do public-private partnerships that they can't do through normal federal mechanisms. We enhance kind of what it is that they are focused on. Now, you asked about how are we funded, kind of versus, what people more might think more traditionally for NIH. We have a small stipend from NIH that we get yearly. It's just enough to probably cover, a small portion of our operating budget. But the vast majority of our operating budget, as well as all the project funds that we use for our side of the funds, meaning the private sector side of the funds, we raise those from various organizations within the private sector. That includes both, industry as people might think it, so corporate donations, but also philanthropic support, so nonprofits, other organizations that have interest in the area. And so it can really be, you know, all industry, all philanthropic, a combination of the both. But again, whatever we feel is the right sort of mix of funding for a particular project.
You're leading a new program at FNIH to advance innovative technologies that model human biology, enable faster, less expensive testing. This is a public private partnership dubbed the Validation and Qualification Network. It's part of the NIH's Complement Animal Research and Experimentation Program. Can you give a sense of who the key participants include?
Absolutely. Obviously, you just highlighted one of the key ones, which is NIH. We are the Validation Qualification Network, or the VQN, is the third pillar under complementary, which is complement animal research and experimentation. But in addition to NIH, we actually have about 14 other US government agencies. So that includes FDA, EPA, NASA, NIST, others coming to the table with us. We also have regulatory and scientific bodies from foreign, governments represented. As far as the private sector, we have pharma companies, chemical companies, other end users. So we really consider those groups to be our end users of what these new approach methodologies are for. We have small company methods developers. So those are the folks who actually develop the NAMs. We have CROs and CDMOs. These folks sometimes, so that's contract research organizations and contract data methods organizations. We have both of these groups that sometimes execute on the tests that have been developed, or sometimes they are also developing the tests themselves. And then nonprofits of various types. So we have some that focus on animal welfare. We have some that focus on better policy around drug development. We have those that are already working in the collaborative space around methods development, and even some that have entire regulatory focus. So it's a really broad tent that we've brought a number of different organizations under to help us kind of move all of this work in a one direction for the field.
How will the partnership work and what will it seek to do?
So overall, you spoke a little bit about the complement in animal research and experimentation or complementary. Um, the whole picture of that, all three pillars together, are meant to develop, standardize, harmonize, and then push towards regulatory implementation, new approach methodologies. In particular, the Validation Qualification Network, or the VQN, will focus in the latter stages of that. So, taking new approach methodologies that have a reasonable amount of validation, perhaps some that have been working in the field, some might be on the market, some might not be. But we're going to take those methodologies and work towards getting them accepted by regulators for use in preclinical packages, whether that be for safety and toxicology in drug development, whether that's safety and toxicology in the chemical company for use in various capacities, or whether that's even drug efficacy for drug testing. But whatever the specific intended use of the new approach methodology, that's what we will be focusing on and pushing that down the way. And to do that, I told you we have all of these different groups at the table. For each project where we select a new approach methodology to develop a pre-competitive project around, we will have not only the methods developers who are working on creating those NAMs, but also end users who will, chime in with how they see themselves using these methodologies, what they think will help them with what they need to do. And then we will also have regulators sitting as general guidance to, help us make sure that whatever it is that we are creating will eventually be acceptable to them, that the data being produced will be of a quality for evidentiary acceptance.
Well, let's talk about the types of technologies that are being explored. I'd like to touch on a few of those and have you give a sense of the potential technologies that might be used. Can you walk us through those, how they work, how they may compare to animal testing in terms of speed, cost, and accuracy, and the challenges they may pose? Let's start with organ on a chip.
Yes, and I'll keep my descriptions of these fairly high level, so hopefully none of the scientists listening will get upset that these aren't probably as detailed as they would hope. But
We'll be forgiven.
thank you. Human cells. So for organs on a chip, you can think about these as human cells enclosed in various types of um, microfluidic chips. And I know that's not the most non-scientific description of these, but it's essentially like silicone or plastic chips that are built to replicate aspects of certain human biology. So, blood flow or cardiac timing, that sort of thing. It just sort of depends on what the chip is intended to do. So the cells selected to be put in there will be dependent on, what the ultimate use of the chip will be. And just as an example, in case we have listeners who aren't as familiar, you could think about this, taking liver stem cells from a human, placing them in a type of microfluidic chip, as we've just discussed, and then being able to have chemicals sort of processed through as if chemicals were flowing through a bloodstream across these cells, and then determine how those chemicals are causing those cells to react. So uh, it is, as you might guess, faster than animal models. You can mass produce these chips, and can do that at a much faster process than, mice can essentially propagate. Uh, and then you can process a lot more chemicals through these, because sometimes, it's a one and done use of a chip, but sometimes you can actually use a chip multiple times for chemical screening or drug screening. And so at scale, these are going to be much lower of cost, right? You're not paying for the housing of the animals, you're not paying for kind of the long term experiments. And once you kind of get something up and running and you can mass produce them, you can, kind of produce these at a fraction of the cost to do everything else. And so as for accuracy, you know, depending on the model and what you intend as the use for that particular model, I think that they can actually be more accurate than animals. They have the advantage of actually being human cells. They have the advantage of being able to replicate certain things now with the technologies that we couldn't in the past. But I will say, I don't want to generalize that there may still be reasons that animal models might be more effective in certain instances.
How about the use of cell lines?
Right. So, 2D culture, which is what a lot of people I think about for cell lines, meaning that you grow the cells kind of on flat dishes and that sort of thing. That's been done for decades. We've used this for a lot in science, especially in drug development. They're less able to replicate kind of full human biology. We don't have the ability to kind of do things the way you would in a microfluidic chip, changing, the pressures or changing the flow of things. But they can be very effective in initial high throughput screening for chemicals and drug tox. Because again, they grow quite quickly. You can do this in mass and, you can test a bunch of chemicals that way. I think, there would be arguments with people within the new approach methodologies field as to whether or not they figure 2D culture is actually part of that. I mention it just because I think for comprehensiveness, what we really start to talk about with cell lines is when you start talking about 3D organoids. So these are cells grown in 3D structures, media that complements the ability to do that. And some of them can actually get to the point of actually producing what looks like small organs within the system that they're grown in. I mean, you could have hearts that literally look like they're beating. You can have complex structures. I'm a cancer biologist, so we've done this a lot in cancer, where you can actually not only have the tumor cells, but you can have vessels and various things growing in amongst them. And that's when you really start to get a better replication of the human biology when they're grown that way. And so, as long as you derive the cells from appropriate cells within a human subject, I actually think they're probably, very accurate, in many cases can be more accurate than animals, or at least as accurate, and they're definitely going to be faster and more cost effective for the same reasons as organ on a chip and cell lines.
Finally, I wanted to ask you about AI models. Where are we in seeing these being able to predict drug safety with accuracy?
Yeah, it's a very good question. I think these probably are not as far along as the in vitro models, just by virtue of, I think, AI and algorithms are just getting better and they're really starting to hit. The biggest thing for these models is the appropriate training data sets, to be able to train, the algorithm on. And so I think we need to do a better job of amassing these types of data sets. There are groups that have begun to do this work within NIEHS for human toxicology studies. HESI and others have sort of started to put these together. But one of the other pillars for complementary is to actually create a data hub and data coordinating center in which we will begin to even better centralize these resources for the training of these. I will say, even despite that, kind of slight drawback that I just articulated, there are groups who are already using AI in combination with either pre-existing human and animal data or actually other new approach methodology data. So there are companies out there. Vivadyne, I know, has been in the press of late, where they're using kind of 3D or organoid screening with a back-end AI algorithm, so they can do all this high throughput screening, and then the AI algorithm actually analyzes all the data in an enclosed system for them. There's a small company called Vile, that they've actually used new approach methodologies, and AI to actually get to first in human studies. Now, I think most of their applications are currently residing within Australia, but again, still an example that these are things that are being used. I think we just need more and better data to continue to train the algorithms. And once we get that kind of data, they should be much more cost effective and much more able to replicate because we're essentially going to be using actual human data or good model system data to do it.
One of the big challenges here is replicating the complexity of human biology. A lot of these alternative technologies may be very good at um, giving you how a drug might impact a particular cell type or system, but how big a challenge does that remain?
It's still a relatively big challenge. Obviously, different models are good for different types of things. I will say the technology is just getting so much better, though. Those 3D organoid models and other types of new approach methodologies that I've mentioned, they're actually experimenting with multi-cell type models, and so some of them even have the ability to kind of fully replicate um, small kind of microcosms of human biology. I'll use again. I mentioned Vividyne earlier. They've been doing a lot in the cancer field where they've actually been able to construct high throughput, well based organoid models that actually include cancer cells. Human blood vessel cells and various types of human immune cells. And it's especially that immune component that is often people feel is lacking in new approach methodologies because it's not something you generally replicate in vitro, but only in vivo. And so, if you can now begin to add in the immune component to these in vitro technologies, you begin to start to replicate things in a much, more consistent fashion to what's actually happening in the human body. Now, given it's still just a microcosm, it's still just one aspect of what's going on, kind of, in the whole of the human body. But I think those types of methodologies are really going to be where the future lies.
About 20 years ago, there was a lot of buzz around the potential of microdosing, giving small amount of drugs to humans as a way to test for safety. I know the FDA and EMA both issued guidance back then, but have we seen this approach advance at all? And is the network expected to explore this?
I think the approach has advanced, although I will admit I'm far less versed in this than others would be. At this particular time, it's not going to be a focus for our particular new approach methodology work, but that doesn't mean that in the future, if it becomes kind of obvious that this is a direction to go, that we could not pivot.
This is a long-term project. Can you give some sense of timelines and how far FNIH plans to take any technology?
Absolutely. So right now we're working on an early phase one, year one, where we are working to put in place all of the necessary infrastructure for the validation qualification network and for selecting our proof of concept pilot projects that we want to work with. So we anticipate this started in June of this year. We anticipate to be done by March of next year and ready to launch those pilot projects in full effect, as well as having the overarching structure of what the VQN will be. And we do hope that overarching infrastructure allows it to be kind of evergreen, and ability to go out as far as we have interest and funding to do so. We already know that as far as the next step, we have the ability for 5 years worth of NIH support for implementation of those projects that we select, both those first four and some additional ones that we'll likely select in year two and three. Uh, we also know that that's probably not quite sufficient amount to support what it is that we'll want to do fully. So we will also be recruiting both financial and in-kind support from the private sector for those 5 years. And then, if we are very lucky, and as I said, we are able to create this evergreen model that we hope we will continue to solicit ideas and concepts for these pre-competitive projects. NIH envisions at least another 5 years beyond this first six that I just talked about. And we hope to actually go well beyond that. We have a particular notion of we're going to be structuring the VQN a little bit after another partnership that we have called the Biomarkers Consortium, where we've worked to move biomarkers down the regulatory development pathway. And we've been doing that for 20 years with over 40 projects having fallen under that umbrella type program. And so we hope that that's what the VQN will eventually be.
There'll need to be regulatory acceptance of any technology. What'll it take to get there and have these new approaches integrated into drug development and accepted in a preclinical data package?
Correct. Absolutely. Regulatory acceptance, quintessential goal of what the VQN will be looking for. We hope that by having the regulators at the table early and often, we have a number of representatives from the FDA, from all spans of the FDA. So that's both drug development, food safety, vet tech, the whole of what they cover, sitting at the table for the steering committee to help us design how we're going to select projects, what projects should look like. We have EPA and EMA and PDMA sitting as well in these types of capacities. We hope that they will then also sit as counselors to the projects we select so that we design the projects such that we are targeting getting the right kind of evidence for these to be accepted by the regulators in the future, and that we're processing the data and reporting it to them in the appropriate kind of standard fashion. So all of that will be part of it. And, you know, what I really think it will take in the long run is, you know, well validated models that can be reproducible across different labs, really put out and used kind of, I guess, not exactly the word I'm looking for, but in a commercial method, meaning anybody could kind of drop in, use these, any lab could use them. And then, a standard reporting of what the, regulators expect to see. Here are the various readouts that need to happen. They need to come to us in this format. And that those last two in particular are what we hope that we can really help and focus on, since we do have our regulatory colleagues sitting at the table. As for, have these already been integrated into drug development and preclinical data package? Some of them have been, yes. Some new approach methodologies have been part of preclinical data packages. Uh, they've been accepted for, use when filing for kind of drug work. Have any of them been fully qualified and fully accepted as standalone by the FDA? That has not happened yet. They've all been kind of accepted in specific contexts of use for specific drugs. There are programs in which we can get the regulators to, as they would say, big Q qualify them, things through programs like iStand, where then anybody could use them, in any preclinical method, as long as they're using it in the context of use that it was approved.
As you think out to the future, how do you see preclinical drug development changing as a result of these new types of technologies that may end the use of animal testing?
I think and I hope that we'll start to see kind of um fail faster kind of model, given that, as I articulated, 90% of all drugs fail in trials. I'd love to see more things not make it out of preclinical stages by use of these technologies that are better replicative of human biology. We can process through faster, that, what we actually end up taking into trials and giving to patients, we have much more of a chance for it to succeed and really show benefit and effect for patients. And I really hope that we can do that on a much more accelerated timeline, because these don't take, weeks to years as the animal models do. We can do them in days to weeks. And the reason that I hope that is the average drug to get to market fully, through a regulator is often 10 years or more. I would love to see that timeline cut in half or even to a third of it, where we could begin to see drugs getting to the market a lot faster than that, because there's so many underserved areas that we aren't getting to for drug development, because we're focusing so much on higher areas of prevalence for drug and disease, that I think if we can start to get some of these going faster, maybe we can start to focus on areas that need a lot more attention.
Stacey Adam, Vice President of Scientific Partnerships for the Foundation for the National Institutes of Health. Stacey, thanks so much for your time today.
Thanks, Danny. Glad to be here.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.